Assalamu alaikum. Hope everyone is fine. Now we are going to discuss the histology of the bone, and we have finished the histology of the cartilage. So in this we discuss the functions of the bone, what is the composition and various types of the cells which are present in the bone, the composition of the bone matrix, what is periosteum and the endosteum, the types of the bone, the differences between the compact and the spongy bone, the process of osteogenesis, and a few of the medical applications. Bone is a specialized connective tissue. It is characterized by a mineralized extracellular matrix. It is composed of cells and the extracellular matrix. The mineral is calcium phosphate in the form of the hydroxyapatite crystals. Bone serves various functions. The basic function of the bone is the storage of the calcium and the phosphate. The bone is the basic unit of the Bone is the basic unit of the human skeletal system, and it provides the framework for and bears the weight of the body. It protects many vital organs, it supports the mechanical movement, and hosts the hematopoietic cells. It also maintains the iron homeostasis. Bone is composed of the extracellular matrix and the cells. The cells which are present in the bones, they are the osteoblasts, the osteoclasts, the osteocytes, and the osteoprogenitor cells. The extracellular matrix is composed of the organic component and the inorganic component. The inorganic component consists of the minerals, in which the calcium phosphate in the form of the hydroxyapatite crystals is the major mineral which is present in it, and the other minerals are also present like the bicarbonates, magnesium, and the calcium and the phosphate in the free form. The organic matrix of the bone consists of the proteins, the collagen fibers bound by the amorphous mucopolysaccharide substance. The bone cells, they are the different types of the cells where they are located. Their location is also different. The osteoprogenitor cells or the osteogenic cells, they are the stem cells for the bone. They are derived from the mesenchyme and they are present just near the periosteum, and these are responsible for the formation of the osteoblast cells. These osteoblast cells, they are the immature cells, they are the bone forming cells. They secrete the extracellular matrix, and once they secrete the extracellular matrix, then they become the almond shaped, and they have the numerous processes on it, and then they are called the osteocytes. The osteocytes, they remain in a space which is called the lacunae, and they maintain the bone tissue. That is, they are responsible for the deposition of the calcium and the phosphate in the bones. The other type of the cells, they are the osteoclast cells. These osteoclast cells, they are the large multi-nucleated giant cells. They have the one edges, the ruffled, which is called the ruffled border, and they are located on our inner side as in the resorption bay. These osteoclast cells, they are derived from the blood monocytes, and they are similar to the macrophages of the blood system, and they are responsible for the resorption of the bone. So these are the functions of the various types of the cells which we have discussed. The osteoclast cells, they release the lysosomal enzymes and hydrolytic enzymes to break down the bone matrix. Now, the bone matrix is composed of the organic and the inorganic part. The 90% of the organic component of the bone matrix, of the dry weight of the bone matrix, is made up of the collagen type 1 fibers, And in this, it's a multi-adhesive glycoproteins, they are also present, like the osteonectin. And calcium binding proteins, they are the osteocalcin and phosphatases. Whereas the inorganic component of the bone is formed by the various minerals, in which the majority of the mineral is the calcium hydroxyapatite in the form of the crystals. And the other minerals, they are the bicarbonate, sodium, potassium, magnesium, and the non-crystalline form of the calcium and phosphate. Now the bone, the outer covering of the bone or the compact bone, it is surrounded by the periosteum. The periosteum is a layer of the dense connective tissue which is surrounding the whole circumference of the bone. Now this periosteal layer has the two layers, the outer layer and the inner layer. The outer layer is a fibrous layer which is composed of the collagen fibers with the typical fibroblast cells, and the inner layer is cellular, and this inner layer, cellular layer, composed of the osteoprogenitor cells. Now you can see here in this photomicrograph, this is the periosteal layer composed of the dense collagen tissue, and these are the fibroblasts, whereas the inner layer is cellular and composed of the osteoprogenitor cells. Now these osteoprogenitor cells in the outer layer, they are called the periosteal cells, and whereas in the endosteum, that is the layer which is close to the marrow cavity and the spongy bone, they are called the endosteal cells. The marrow surface of the compact bone and the spicules of the spongy bone, they are lined by the single layer of the cells, that is the endosteal cells. The endosteal cells, they are also osteoprogenitor cells, and they are also capable of becoming the osteoblasts. The two names, that is the periosteal cells and the endosteal cells, they refer to their different locations. Both function as the osteoprogenitor cells. Then there are the Sharpey's fibers, which are also called the perforating fibers. They are the bundles of the collagen fibers, which are derived from the periosteum, and they penetrate into the bone matrix at an angle. They are continuous with the outer circumferential lamellae and the interstitial lamellae of the bone matrix, and they hold the periosteum with the bone matrix. Now, the bone is classified histologically into two types, that is the compact bone or the lamellar bone. It is also called the dense bone, and the spongy or the cancellous bone or the trabecular bone. Now, this diagram showing you the long bone in which the diaphysis is composed of the compact bone, and the epiphysis or the metaphyseal plate is composed of the spongy bone. Now you can see here the compact bone along with the inside is the marrow cavity surrounded by the compact bone. And this is the epiphysis, and you can see here the bone spicules in which there are these marrow cavities filled with the marrow in the living. So compact bone and the spongy bone, they are found in specific locations. So in long bones, most of the thickness of the diaphysis is made up of the compact bone with a small amount of spongy bone facing the marrow cavity. And the ends, that is the epiphysis of the long bones, they consist mostly of the spongy bone covered with the shell of compact bone. The flat bones of the skull, they have a middle layer of spongy bone which is sandwiched between the two relatively thick layers of compact bone. So this is the diagram showing you the structure of the compact bone or the dense bone or the lamellar bone. So why it is called a lamellar bone? Because it consists of the collagen fibers which are arranged in the layers of the bone. They are called the lamellae. So in the outer part, they are arranged in below the periosteum. It is layer, these lamellae, they are parallel to each other, which are called the outer circumferential lamellae, And similarly, the close to the end ostium, they are also parallel to each other, which is called the inner circumferential lamellae. Whereas in the substance of the compact bone, these lamellae, they are arranged in concentric whorls, and these concentric whorls, they are arranged around the central canal, which is called the Haversian canal. And this Haversian canal has the arteries, veins, and the nerves which are passing through it. So these concentric lamellae, along with their Haversian canals, and in which the osteocytes, they are embedded in these concentric lamellae, and they lie in their spaces which are called the lacunae. So this system is called the Haversian system. And one Haversian canal, along with its concentric lamellae, it forms the one osteon. And the substance of the compact bone is made up of the various numerous osteons. Now you can see here, this is the one osteon and this is another osteon, and in between the two osteons is the interstitial lamellae. Now these interstitial lamellae, they are the remnants of the previous osteons. Now you can see here in the longitudinal section of the bone, this is the transverse section and this is the longitudinal section. Now this is one Haversian canal and this is another Haversian canal. The two Haversian canals, they are connected with each other in an H-shaped manner, which is called the Volkmann's canal. Now you can see this Volkmann's canal in the longitudinal section only, and in the transverse section you can see the Haversian canals. So you can see here the section of this bone here, and these osteocytes which are present in the lacunae or the space, and these osteocytes, they have the numerous processes which run in the canaliculi. These processes, they are responsible for deriving the nutrition from the central Haversian canal arteries, and they provide this nutrition to the distant osteocytes in the distant lamellae. So these canaliculi, they connect the lacunae with one another and with the Haversian canal, and the canaliculi contain the processes of the osteocytes which communicate with one another via the gap junctions, and thus the nutrients and the other substances such as the hormones, they can pass from the blood vessels in the Haversian canal to distant osteocytes. So these osteocytes, they are involved in the bone deposition. Now this is the photomicrograph with the special stains of the to show you the compact bone. To stain the bone, we have to first to decalcify to stain the bone. Now you can see here these the concentric whorls of the concentric lamellae around this central Haversian canal, and you can see appreciate these osteocytes in these lamellae, and these are the processes of the osteocytes running in the canaliculi. These processes, they are reach their central Haversian canal. Now, this lamellae, which is just close to the Haversian canal, for example, if we name it a lamellae 1, the osteocytes in the lamellae 1 will withdraw its nutrition from the Haversian canal, and then they will pass it to the lamellae 2 osteocytes through these processes in the canaliculi, and so on to the further areas and the uh, concentric whorls which are away from this Haversian canal. So this is also showing you the Haversian systems, or this one Haversian system or the one osteon, and the compact bone is made up of the various osteons or the Haversian system. Now this is a diagram showing you the comparison of the compact bone or the immature bone or the forming bone. So you can see that in the compact bone there are the concentric whorls, lamellae, and they are concentrically arranged, whereas in the immature bone there are the spicules or the trabeculae of the bones in which these osteocytes, they are embedded in the bone matrix but not in the concentric whorls. And these are the marrow spaces in which the blood vessels, you can see here in the marrow spaces, and this is the marrow cavities filled with the marrow. So the spongy bone is composed of the bone spicules. They are also called the trabeculae. Therefore, the spongy bone is also called the trabecular bone or the cancellous bone. It is of varying, these trabeculae or the spicules of the bones, they are of varying shapes and the sizes. The spaces between the spicules, they are filled with the marrow. The composition of the spongy bone, the cells and the matrix is the same as that of the compact bone. However, in the spongy bone, the lamellae of the collagen, they are not arranged concentrically around the central Haversian canal, but they run parallel to one another. So osteocytes, they sit in the lacunae between the lamellae. So this is the compact bone versus the spongy bone. You can see here the differences of the compact and the spongy bone. This is also the photomicrograph showing you the outer circumferential lamellae just below the periosteal layer, and then is the bone tissue, and then is the inner circumferential layer, and then is the endosteal layer, and this is small amount of the spongy bone which is embedded over it. This photomicrograph showing you all the components of the bone tissue, that is the bone matrix and the cells, the osteoblast cells, which are the immature cells, and they are present on just on the edge below the periosteal layer. And these are the osteocytes, which are bone cells, which are present in the spaces called the lacunae, and these are the marrow spaces filled with the marrow with all the stem cells. So the process of the bone formation or the osteogenesis process is of the two types. That is, the bone develops in the embryo by the two distinct processes, that is the endochondral ossification and the intramembranous ossification. In intramembranous ossification, the bone development is from the connective tissue mesenchyme and there is no previous cartilaginous model is formed. The mesenchymal cells, they differentiate directly to the osteoblast cells, and these osteoblast cells, they produce the extracellular matrix around them, and which soon this extracellular matrix is soon calcifies. And this, when calcifies, it calls the osteoid formation, and these numerous ossification centers, they are formed, they anastomose and produce a network of the spongy bone that consists of the thin rods and the plates on spines, which are called the trabeculae. Now the osteoblasts then become surrounded by the bone in the cave-like lacunae and become the osteocytes. And once the osteocytes, they are formed in the lacunae, they establish the complex cell-to-cell connection connection through these canaliculi. So this is a photomicrograph showing you the growing bone spicule, and this is an electron micrograph showing you the active bone formation, the osteoblast cells, the osteocyte cells, and these are the different types of the cells. Now, the other form of the osteogenesis process or the ossification is by the endochondral ossification. Now, in endochondral ossification, mostly the bones of the body, they are formed by the endochondral ossification. And in this, a temporary hyaline cartilage model, it is formed and then it is later on transformed into the bone. So it grows both by the interstitial and the appositional means. And you can see here, this is the growing cartilage or the growing bone formation, and you can see here the different zones. So this zone is called the zone of the reserve cartilage. This is the zone of the proliferation in which the cells, they proliferate, and the zones of the hypertrophy. And then later on, this is the zone of the calcified cartilage, where the cartilage calcifies, and then is the bone formation takes place in the calcified areas of the cartilage. And then this is the zone of resorption, where the extra bone is resorbed by the osteoclast cells. So these are, you can see the osteoclast cells which lie all in the resorption base, and these resorb and remodel the extra bone. So the chondrocytes, they divide, hypertrophy, enlarge and mature, and the hyaline cartilage model, it begins to calcify. So this is all of the hyaline cartilage, in which there are the different zones of the cartilage. And when the zone, this cartilage model calcifies, it forms the bone. As calcification of the cartilage model continues, the diffusion of the nutrients and gases through the calcified matrix decreases. So consequently, the chondrocytes, they die, and the fragmented calcified matrix, it serves as a structural framework for the deposition of the bony material. Now you can see here the endochondral ossification of the long bones. First, you can see that this is the hyaline cartilage, the whole of the bone in the embryonic life. It is formed by the hyaline model, and then there are the primary centers of the ossification. They first appear for the diaphysis and in a collar-like fashion, and then this collar-like fashion, and later on it is calcified. The central cartilage is calcified, and along with it a collar or the bone is formed in a circumferential manner. Then you can see here that the this calcified matrix is vascularized by the blood vessels, and there are areas of the calcified cartilage or the edges, and the epiphyses, they remain in the hyaline cartilage. Then later on, these ossification centers, they grow from the diaphysis towards these metaphyseal plates. The secondary centers of ossification, they appear for the epiphysis, and later on they also vascularized. So these vascularized and calcified. So later, slowly, the whole bone is formed in this hyaline cartilage model. And then these epiphyseal plates or the cartilage, they are regressed and united with these secondary centers. They are united with these primary centers by the age of the 25. So few of the medical applications which are very common and commonly asked SEQ questions, like the osteosarcoma. So any tumor arising from these osteoprogenitor cells, it is called the osteosarcoma. Now, in the bones, the tumors of the bones itself, they are uncommon, but the metastatic tumors which arise from the other organs and they metastasize in the bones, they are mostly common. For example, the cancers from the breast, the lungs, the prostate gland, the kidneys, and the thyroid gland. Then another term is used with is called the osteopetrosis, and this is commonly asked SEQ question. So it's a genetic disease in which there is heavy bones characterized by the dense heavy bones which are called the marble bones. And in this, the osteoclasts, they lack the ruffled borders. Therefore, they are defective and they are unable to resorb the bone. So the unwanted or the extra bone is present, which later on forms the heavy bones or the marble bones. Then osteoporosis. The osteoporosis is a common disease of the females after the postmenopausal age, and is an imbalance in the skeletal turnover, so that the bone resorption, it exceeds the bone formation. So this leads the calcium loss from the bones and reduce bone mineral density. Then another term used is the osteomalacia. The osteomalacia is the mineralization in the when the mineralization is impaired in the adults. Then osteitis fibrosa cystica in which increased osteoclast activity results in the removal of the bone matrix and the fibrous degeneration. The osteogenesis imperfecta or brittle bone disease, it is referred to a group of related congenital disorders in which the osteoblasts they produce deficient amounts of the type 1 collagen or defective type 1 collagen due to the genetic mutations. So it leads to the significant fragility of the bones. Then rickets, you are familiar with the rickets. It's the calcium deficiency in the children and the bone matrix does not calcify normally and the epiphyseal plates can become distorted and there are the bow-shaped bones which are present. So these medical applications, their names should be familiar with you and they are often asked in the SCQ questions. So this is all about the bone tissue. So we'll discuss later on the another topic or the another epithelial tissue of the body, that is the muscular tissue. Thank you.